This week we're on to a new topic which is on bacterial two-component system or TCS. So what is a two-component system? So two-component system is, as the name says, two components. So it's a coupling mechanism, okay, which requires or required for basic stimulus response. So it is a form of mechanism for sensing and responding to changes, for example, various environmental conditions, whether it's cold, whether it's hot, for example. And critical function, of TCS, is basically in signaling, for example, for communication, for adaptation to environment, for pathogenesis, mutagenesis, and so forth. So a lot of time TCS is unique to prokaryote, but it is also available in some eukaryote, for example, in plant. So don't be surprised if you heard of two-component system in plant, because certain plants do have two-component system as well. But of course, this is mostly for prokaryote. So what is the value of TCS? Higher potential of TCS protein as antibiotics to sustain or for the bacteria to survive. So this system, again, as the name says, comprised of two components. So it basically has two proteins. One which is called a sensor kinase, or histidine kinase, HK. Another one referred to as a response regulator, double R. Now, each and every one of them, have their own function. Basically, should be switched, but never mind. So histidine kinase sensor basically has a conserved kinase core which act as a transmitter domain plus a specific sensory domain. So basically this is your histidine kinase, whereby the response regulator is or has conserved receiver domain plus specific effector domain, just to give the outside or output signal, or how to react, to the transmitted sensor. Now, for the sensor kinase, or the histidine kinase, this is more or less the basics of it, or the characteristic. So it is cytoplasmic, or membrane, and its function is to detect or senses specific environmental signal or stimulus. Once it basically detects or sense this stimulus, it will activate kinase activity. Plus, it will also phosphorylate a conserved histidine residue. So once it has autophosphorylate, phosphorylated group will be transferred to cognate response regulator for activation. So this is more or less how the phosphotransfer reaction occurs. So the phosphoryl, the explanation is basically done down here. So the phosphoryl group is transferred to the conserved histidine kinase side chain. As you can see over here. So what happens is that later on it will be transferred to the response regulator. So that the response regulator could then dephosphorylate and give the final reaction. For example, hydrolysis reaction. So the response regulator characteristics, it is cytoplasmic, or available in the cytoplasm. So it is actually a DNA binding protein, and basically it is a form of transcription regulator. So it could regulate transcription. So what happens is that once it receive a signal, so it will phosphorylate the signal by the sensor kinase, histidine kinase. So it will respond to the stimulus by expressing target genes accordingly, whether to turn it on or to turn it off, depending on the kind of stimulus that it received from the histidine kinase. So all in all, this is how the two component system looks like. You have your histidine kinase over here, and you have your response regulator over here. So it will receive the signal, and then it will basically autophosphorylate. So the signal is being conveyed to the response regulator. The response regulator will dephosphorylate and give out the output, whether the gene should be turned on or turned off. So this is basically more picture, so that you could get hopefully get the idea of how the system works. So histidine kinase is actually in a form of dimer. So you have two catalytic domain, basically on the sensor domain. And what happens is that energy conversion is being done to here during autophosphorylation, and the same thing happens in response regulator as well. Now, basically, in form of genomic distribution, okay, certain systems have multiple regulatory elements. For example, E. coli. Okay, and I've put here, okay, as you can see, in E. coli there are more than 50 TCS alone. For each bacteria, they usually have more than one TCS, and the name varies one to another. So you can see, okay, over here, for example, in E. coli, okay, there are more than 50 TCS. Okay, the T histidine kinase, okay, 5 of them are hybrids, and 32, okay, response regulator. So the type of histidine kinase and response regulator could be similar, but okay, just that, okay, how they work, okay, or how they react, okay, upon receiving the stimulus, okay, and giving out the output, okay, is different. In Syncoctis species, okay, about 80, there is none in Mycoplasma so far. In Bacillus subtilis, 70. Haemophilus influenza, 9. Okay, Helicobacter pylori, the one that gives you, okay, cholera, okay, basically around 11. And since there are a lot of TCS, okay, there is a potential for crosstalk. So what are crosstalk? So we will see, okay, in a while. So how do two component system, okay, regulate the gene expression? So this is or whatever that I've put here basically are just some of the examples. Okay, for example, regulation of toxin gene expression, and this is basically quite important, and hence, okay, it would help, okay, with the bacteria or the virus itself. For example, okay, in Bordetella pertussis, okay, so produces pertussis toxin. Okay, so this is the system BVGS and BVGA. Okay, in Clostridium, okay, Paphidium for alpha toxin. Okay, this system, Staphylococcus aureus, and also pyrogenes. Another thing, okay, or another function, okay, of the regulatory mechanism, okay, is a regulation of other virulence factor, okay, such as like the RN4P in Enterix bacteria and also in Salmonella. So just to give you a rough overview, okay, of how it is being used. And as you can see from the names over here, okay, there are lots of TCS available, just that probably some of you are not too familiar with the two-component system. So we get to the roles of the two-component system. As we know that histidine kinase, okay, would sense, so its role is to sense, respond, and adapt to a range of environment stresses, okay, the one that gives stress, and of course different growth condition depending on the environment. So this would include, okay, the response, okay, to nutrients availability, redox reaction, osmolarity, quorum signals, antibiotics, temperature, chemoattractants, pH, and of course many more. Everything that is related, okay, to the development, growth and development of the prokaryote itself. So titer regulation, okay, usually is being administered to prevent unwanted crosstalk by bacteria, okay, with more than one TCS. Now, what do I mean by crosstalk, okay, just now, okay, and if you know the meaning of crosstalk generally, okay, is that, okay, sometimes, okay, there would be interference, okay, from one component system to another. So when there is interference or crosstalk, okay, which means that it would play around or play havoc with the expression of the gene, okay, or with the control of the adaptation. So one example, okay, of two-component system is actually quorum sensing and virulence, which is in reaction, okay, to viruses. Do you guys remember about quorum sensing? I'm not sure whether you learned this during your undergrad or not, but during my undergrad, I remember learning about quorum sensing. So quorum sensing, for any one of you who don't remember or have forgotten, is basically a mechanism bacteria use to assess their population density. This is whereby, okay, when they would assess the environment when they need to grow or overpopulate, okay, or basically make sure that they would survive. So it would ensure sufficient cells present before initiating response. And how do they do that? They have specific autoinducer molecule, okay, for example, okay, as you can see over here. So autoinducer diffuses freely across the cell envelope, and once it reaches high concentration inside cells, then only, okay, it will basically tell us that, okay, you can just, okay, populate or overpopulate now, okay, because there are many cells, okay, for you to grow, okay, and overpopulate. So in this case, okay, for quorum sensing, it binds to specific activator and triggers specific gene transcription to make sure that they could survive, for example, okay, over here. So for growth and establishment, okay, so they would need, okay, to make sure, okay, that pathogenic factors, okay, are not there, okay, and there are, for example, pheromones, okay, to help them to grow. Okay, in terms of recognition of cell density, so whether the cell has enough density or not. Okay, so if they need to reduce its density or whatever, so current cleansing is being used, and especially for specific gene expression. Okay, so for example, over here, okay, is whereby, okay, when there is infection, okay, so they will attack, okay, to make sure that they could survive over there, and this is the survival of the fittest prokaryote style. All right. Okay, so this is the basic on the two-component system. Move on to the next video on the details of two-component system and the extra feature of two-component system for some prokaryote.